Welcome back to my course Aspects of Biochemical Engineering. And last couple of lectures we try to discuss the different numerical problems on the kinetics of substrate utilization, product formation and biomass production using microbial cells. Now today this is the last part of the numerical problems that we are going to discuss today. And in last couple of classes we try to tell you that how to analyze the batch system, how to analyze the chemostat, how to analyze the plug flow reactor, and then also we talk about some inhibition problem, substrate inhibition, product inhibition, all this we try to discuss. And also we try to find out the procedure, or that techniques through which we can find out the kinetic constants like maximum specific growth rate, saturation constant, true growth yield and the maintenance coefficient. Now today this lecture we try to cover a little bit different, but what we want to tell here that we already discussed how the batch data can help you to find out the data for the chemostat process. As for example, from the batch analysis we can find out that at a particular dilution rate what will be the cell mass concentration in a chemostat. We can easily find out from the batch process. Then we try to discuss the different type of systems. As for example, that if we use the multiple reactor in series, how you can find out the cell mass concentration, because we have seen before that in case of cascade type of fermenter that you can easily determine the substrate concentration in different reactors, cell mass concentration in different reactors by doing the substrate balance, cell mass balance. But for when you do that, then we have come across different type of quadratic equations, then you still have to solve it. But this problem, when the problem that I shall discuss, I shall show you how graphically you can find the cell mass concentration after n number of reactors, and also what will be the pattern that we have in case of dioxic growth of the cells. This is the dioxic growth. We know it is special type of growth because if the media contains more than one carbon source, some cases until and unless one carbon source is totally exhausted, other carbon source will not be utilized. So after this, again we shall go for the chemostat process to find out what is the time required for a particular amount of substrate conversion and all these things by using Zymomonas mobilis for a particular product formation. We try to find out, and after that, at the end, what I am planning to discuss that how this Luteking Pirate model we can use just to find out that at what dilution rate we can have the maximum rate of product formation. And also from the Dmax value, we know Dmax equal to mu max into 1 minus root over Ks by placed by S0, how you can find out the maximum dilution rate at we will get the maximum rate of cell mass concentration. So when we say, let us start with this first problem that we have, and if you look at this problem, the problem says that mass balance for component C in batch or plug flow reactor and chemostats are this. This for the batch and plug flow reactor, dC by dt equal to Fc, that is the function of C, and in case of steady state chemostat, dC0 minus C equal to minus Fc, because we have seen in case of chemostat process, because in case of batch or batch process, T batch, in case of T batch, what we can write, T batch equal to what we have seen, that minus dC by minus RC. Am I right? This is same as your tau's Planck flow reactor. Planck flow reactor, this we have seen, but in case of tau CSTR, we have observed this is equal to C0 minus C by minus RC, the rate of degradation of C. Okay. Now this is exactly that they try to find out that this is D into C0 minus C function of C, and this is like this. Now since the Fc is available from the by differentiation, a plot of Fc and versus C can be constructed for the batch data, and equation 2 indicates the plot of C0 minus C versus C will intersect Fc plus C as C star correspond to the solution C. Now what does it mean actually? Let me explain this. So in a batch process, so suppose when you talk about the batch process, now in the batch process how you operate with, this is the reactor that we have, and it is rotating like this. So time to time you draw the sample. So what you can do, we can at different time we can have different substrate concentration and different cell mass concentration. Am I right? And then we can find out dx by dt. And I told you dx by dt, how we can find out dx by dt equal to x n plus 1 minus x n minus 1 divided by 2 del t. That is the n is the number of sampling point, 1, 2, 3, 4 like this. Now in this particular batch data, what we can do, we can plot dx by dt versus x. Am I right? And then we have the plot like this. Now what this problem says, in the CSTR what you have, dC0 minus C equal to minus function of C. Am I right? Now what is the slope? Slope stands for, this is, if you consider this is 0, then this slope with the tan theta is equal to what you have, this this altitude versus base, that is dx by dt divided by x. Am I right? And this is equal to mu, and we know in case of chemostat, under steady state condition and sterile feed, mu equal to d. So this that means this is a particular dilution rate. So we can have different dilution rate, we can have different so they instead of x they are considering C, this is replaced by C. So this is indicating the C star, this is indicating the C star. So this is how in this problem we can make the correlate the batch process with the chemostat process. Now the problem is that considering the logistic equation dx equal to by dt equal to mu x 1 minus x by x max, mu equal to 1 our inverse, x max is the maximum cell concentration 10 gram per liter, and x0 feed is 0. Solve the x star graphically for the case when d equal to 1.5 our inverse, 0.75 our inverse, 0.25 our inverse, using the method discussed above, and verify your results directly from the direct solution analytically. So what we shall have to do, we shall have to solve it both graphically as well as analytically. So let us see how we can solve it. And other portion of the problem is that for the fermenters in series, the outlet from one chemostat is the inlet condition for the next, and so graphically how would you evaluate x3 dash, the biomass concentration of the third CSTR in 3 reactor cascade with a overall dilution rate of 0.75. So we shall have to find out that, suppose we have, this is one CSTR and this is another CSTR, and this is the CSTR that we have. They are they are connecting to each other. So here we have x1 start, here at the steady state condition x2 start, and here you have x3 start. So we shall have to find out what is the x3 start when d equal to 0.75 hour inverse. This is the problem that we have. Now last problem, that inspect the data from dioxide growth carefully and differentiate it graphically and plot dx by dt versus x. How does this plot differ from the same of simple logistic form? Sketch using the graphical design procedure above a solution of 5 tanks in series which consume all of the glucose and most of the second carbohydrate as well. So we know that dioxide growth means what? Dioxide growth means if you have the cell mass concentration with respect to time. So it is going like this, and then until unless first carbon source is totally exhausted, the second carbon source will not be utilized. The nature of dioxide growth is like this. So during this dioxide growth, what will be the nature of the graph if we plot dx by dt versus x? What should be the nature of plot? That we shall have to find out, and then we shall have to find out graphically that you show me under what circumstances if we use 5 tanks in series, it consumes not only the first carbon source, but also it will consume the second carbon source. Now this is the solution that we have. The logistic equation that has been given, dx equal to mu x 1 minus x by x max. Now what we can do here, all values are given. The mu value is is a 1 hour inverse, x max is 10 gram per liter, and x0 is. So we shall have to we shall have to do, we shall have to plot dx by dt. We shall have to plot dx by dt versus x. Am I right? So what we can do, we can prepare a table. In the table we have 2 columns. One is x, another is dx by dt. So this dx by dt will come here. This x will be here. Now we know maximum value of x max is 10 gram per liter. So we can assume this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We can assume a respective value of dx by dt. We can find it out. And if we have this, then we will be having this kind of plot. You know, this plot we will be having. We can have a plot like this. And once we have this plot, then we shall have to find out that that at different dilution rate, one is d equal to 1.5 hour inverse, 0.75 hour, and 0.25 hour inverse. What will be the x value? Now, if you considered d value is 1.5, we find that tan theta is a little bit high, so it will not touch the curve. So we can assume that this is the situation where the cells will be washed out from the system, because this we can easily visualize from this that this is this from this particular figure, that this is there should be no cell present in the reactor. That when we do it analytically, then also we can justify how it happens. But when we have 0.75, then it touches the curve here. So your cell mass concentration will be 2.5. Now when you have 0.25 dilution rate, then it is 0.75. Now we know 1 by d equal to HRT. Am I right? Now that means if the dilution rate is high, that means you are allowing the cell to reside in the reactor for smaller period of time. And if the d value is low, that means you are keeping the cell for longer period of time. Then naturally you have more cell growth in the reactor. Now next problem that we have, that that we have this analytically we can solve like this, that this is the logistic equation that we have. Now we can bring this side, and we can write mu equal to d. Then we have the correlation like this, and then we can find out the expression for x equal to x max equal to, we can write in other way, we can write in this form. Then we have this equation. We have x equal to 10, x max is the 10 value, 1 minus d by 1. Now, in this equation, if we put the value of d 1.5 directly in this equation, is value is x value is coming about -5, which is not possible. Am I right? So this is, that means this is kind of a situation when there should not be any cell present in the reactor. But 0.75 it is 2.5, and 0.25 it is 7.5. So in the graphically also we find almost the same results. So there is a imbalance between the graphical and the analytical solution of this particular problem. Now, if you look at the second case, that what is saying that same curve we have now 0.75. So as I told you, they try to use the cascade fermenter. That means 3 CSTR that will be in series. They are connecting to each other. Am I right? And they have the same dilution rate, d equal to 0.75 hour inverse. Now here, if you look at this angle is 0.75, this is 0.75, and this is 0.75. So this same angle that we maintain, that means 0.75 dilution rate. Here we have the value of x1, and then here we have the value of x2. This is the point that we have, and here we have the value of x3. So graphically we can easily find out that after third reactor in series, what will be the concentration of the reactor under concentration of the cell mass under steady state condition is very easily find out. Now, if you go to the last part of this problem, what is saying that we are talking about dioxic growth of the organism. Usually the logistic growth is like this, that that exponentially grow like this with respect to time. But in the dioxic growth, I told you that if the media comprises of more than one carbon source, some of the organisms has the characteristics that until and unless the first carbon source is fully utilized, the other carbon source cannot be utilized due to the nature of metabolism of the particular organism. So this is the nature of dioxic growth. Now, if we write this dioxic growth in case of this dx by dt versus x, we will get the nature of the plot is like this. So this will be the nature of the plot. In the logistic equation we have, and dioxic growth we have this kind of plot that we have. Now what we can do here, now they are saying that we shall have to run 5 reactors in series. So in series, so, and we shall have to prove under what circumstances all the carbon source will be exhausted, first as well as second. Now let us take the example. Suppose we maintain this dilution rate, then we can have this, we can have this, we can have this, then we can have this, we can have this. So if you look at, this is x1, this is x2, this is x3, this is might be x4, the x5. Am I right? So we assume the most of the substrate consumed here. Am I right? So so what we shall have to do? We shall have to, but then we shall have to change the curve little bit more, because that d value will decrease the hydraulic detention time. We shall have to increase little bit, so that if you do like this, so we can see. So you will find that x5, when most of the carbon source will be exhausted. So this is the trajectory how we can find out that under what circumstances all the carbon source will be exhausted. So we can easily find out if you do the analysis of the particular process. Now this is here we have the solution. I hope you understand that this is like this 1, 2. This is x1, this is x2, x3, x4, and x5. So when it comes here, we can assume that most of the carbon source will be exhausted. This is how we can solve this problem. Now let me go to the second problem, which is also very interesting. We know Zymomonas mobilis, that is used for the alcohol fermentation process. Now in Zymomonas mobilis, cells are used in a chemostat of 60 cubic meter fermenter. 60 cubic meter means 60,000 liters. Am I right? Now, yield of biomass is about 0.06 gram per liter. So, we know that organism under anaerobic conditions they produce the alcohol. Am I right? And under anaerobic condition the yield cell is very less as compared to aerobic process. Aerobic process cell yield always very high. So, that is why you see this is 0.06 gram of cell per gram of substrate consumed. The product yield is 7.7 gram per gram. The maintenance coefficient 2.2 gram per liter hour. Then specific rate of product formation is 3.41 hour. This should be our inverse. You can make little bit correction. The specific rate of product formation unit should be our inverse, and because specific rate of product formation is what, dx by dp by dt. Am I right? So, this is also gram per liter, this is also gram per liter. So, what will be, this will be time inverse. The maximum specific rate of the growth of Zymomonas mobilis is 0.3 hour inverse. The feed contains 12 grams per liter of glucose, and case of the organism saturation constant is 0.2 gram per liter. What is the flow rate is required to the steady state condition. Concentration of the substrate will be 1.5 gram per liter. So, S value that will be under steady state condition is 1 gram per liter, 1.5 gram, that we shall have to find out. So, first we shall have to find out what dilution rate we have this value, and if we know the dilution rate equal to what, F by V. Am I right? Now, we know that our volume of the reactor. So, if you put the volume of the reactor, we can, if you know, find out the dilution rate, we can easily find out the flow rate. So, this problem can be easily solved, and then next part of the problem, at what dilution rate, at the dilution rate of A, what will be the cell density. So, x equal to what? X equal to usually x0 plus y, x0 minus s0 minus s. So, here we have, if we consider the sterile feed, then this should be equal to 0. Then y x by s, the cell yield already given here, s0 value we know, that is, and then we can find s also we know. So, we can easily find out the cell density, that what flow rate the concentration of ethanol is, what at that flow rate what will be the concentration of ethanol produced, that we have given the yield of the product, that is ethanol yield, for 7.7 gram of ethanol per gram of cell mass. So, we can easily find out, and I told you that whenever we try to solve any kind of problem, it is always recommended that you should write all the parameters. So, here these are the parameters that has been given, you please write it down. Then let us see that I told you that how to solve it. So, this is the equation that we have. Then we have this is D equal to mu max under steady state condition and sterile feed. This sterile feed means x0 equal to 0. Then mu equal to D. Then we can write mu max s Ks plus s. Am I right? Now, in this problem, that all values are mu max value we know, S value you know, Ks value you know, we can find out the dilution rate. Now, dilution rate equal to F by V, and so F equal to D into V. So, your flow rate will be 15.6 cubic meter per hour. So, it is very easy to solve it. Now, once we find out that, we shall have to find out the cell density. How we can find out the cell density? Now, if you do the substrate balance, the rate of input where plus the rate of generation of substrate equal to rate of output of the substrate plus rate of consumption of substrate or accumulation of substrate. Now, under steady state condition this will be 0, and in case of substrate balance this will also generation will be equal to 0. So, what we can write that here we can write this is equal to 0, and this is Fs is the this is Fi is the incoming initial substrate concentration, this is final substrate and ds by dt that is contributing for 3 different purposes. One is for cell growth, another is product formation, and there is maintenance of the cells. So, this can be written as mu by y x by s, qp by y p by s. What is the qp? I told you qp is the specific rate of product formation, dp by dt. Am I right? So, if you divide by dp by ds, then what you will get ds? This ds, the dp by dp will cancel, then it will be ds by dt, the specific rate of substrate conversion. If you multiply by x, and you will get the amount of substrate utilized for the product formation, and if you consider multiplied by volume, you will consider whole volume. So, if you consider this, you can find out a equation for the x is the final cell mass concentration. So, then under steady state condition mu equal to d, if we assume that mu can be replaced by d, and then equation will be like this, then if we since the product is directly linked with energy metabolism, the above equation may be related is like that. We can assume that there is no product formation here, then the mostly that the substrate goes for cell mass formation, then the equation will be like this, and we can find out the what is the final cell mass concentration. We can find it out. Now, lastly, if you do the product balance, say as similar to your substrate balance, we can have this equation: rate of product input is F into Pi. The product generation, product formation take place dP by dt into V. This is the product output F by V. Consumption will be 0, and also accumulation will be 0 under steady state condition. Then if we put Pi equal to 0, divided both sides by V, then by V if you divide, then what you will get? You will get that the equation you will get that this equation I can write like this: dP by dt equal to P. Am I right? And dP by this equation I can write Qp into x by d. D is the dilution rate, because if you divide by P, this is P by P, F by V is there. So, here F by V is the dilution rate. So, this we can bring it here. This D we can take it here. So, and what is Qp is the specific rate of product formation dP by dt. So, this is dP. We can easily multiply that. We can then we can write this equation like the Qp. This is so dP by this, if it is the Qp, then I can write the dP by dt equal to Qp into x. So, that is what is written, and d will be coming from here, and then we can come across this equation. We can come across this equation, and we can solve this and find out the product concentration. So, it is not very difficult. Now, last problem, as I told you that this is with respect to Ludeking Piret model. The kinetics of the microbial growth and substrate and mixed growth associated product formations of the chemostat of the equation. This is the, we know this is the rate of cell mass formation, rate of substrate degradation, rate of product formation with respect to Ludeking Piret model, and these are the different data that is been given in this problem. Okay, alpha value, beta value, S0 value, everything is given. That determine the optimum dilution rate for maximizing the product formation, and determine optimum dilution maximizing the cell mass formation. So this can be solved very easily, because what we can do, we can write that in this equation we know that dp equal to, this is equal to dp by dx, dp by dt equal to alpha dx by dt beta into x. This we can write alpha mu plus I can take x common, then it will be 1 by x dx by dt, that will mu. So alpha mu, so this will be alpha mu and plus beta into x. So dp by dt I can write in this form. So in this equation we can, all values are given, just to put the different dilution rate, we assume different dilution rate, because d max value is given. So we can assume the d value less than that, 0.1, 0.15, 0.2, 0.3. So respective s and s and x value we can calculate, and we can calculate the qp value, that then we can find out the d into p value, that is equal to qp into x, that is, this is equal to dp by dt. Am I right? Now if you plot, this is equal to dp by dt. Now if you plot dp by dt versus d, so we will be having this kind of plot. Now at this dilution rate you will get the maximum rate of product formation. So we can easily find out optimum dilution rate at which you will get the maximum rate of product formation. Now in case of cell mass formation, that we know, this we have done before also, that here also we can find out at different dilution rate what is the s value and x value we have already calculated, and if you know the x value you can multiply by d and x by what is this, dx by dt. Am I right? So we can, if you this is equal to dx by dt. Now if you plot with respect to d, we will also, you will get a dilution rate when you will get the maximum value of dx by dt is maximum. So that is here also. But so both we have seen both the product formation and the cell mass formation, we required maximum, we required the at the same dilution rate, well 0.6 our inverse. So in this particular lecture I try to discuss that critical issue with respect to that how batch data can be used for extrapolating to find out the cell mass concentration in a chemostat, and how the cascade reactor, the cell mass concentration of the cascade reactor can be determined graphically, how dioxy growth can be analyzed, and then we find out, we try to solve some kind of chemostat process, and finally we discuss at what dilution rate you will get the maximum product formation and also maximum rate of cell mass formation. Thank you very much.